this session, we'll talk about muscle contraction. Here are the learning objectives today. By the end of this session, students should be able to describe events that occur in excitation contraction coupling in skeletal muscle, explain the molecular mechanisms of muscle contraction, predict the consequences caused by abnormal excitation contraction coupling. Let's start with a case. Miss Lee, a 32-year-old woman, went to the hospital with difficulty chewing food and tired arms while combing her hair. Symptoms disappeared after a brief rest. She's diagnosed with myasthenia gravis and is started on neostigmine, an acetylcholinesterase inhibitor. Questions: why did she get tired when chewing and combing her hair? And why is neostigmine effective in relieving her symptoms? We'll come back here before we finish our discussion today. A muscle is made up of individual cells called muscle fibers. Set longitudinally within the muscle fibers are bundles of myobrils. Each myobril contains jagged, thick, and thin filaments. They are arranged longitudinally in sarcomeres. The repeating units of the sarcomeres account for the unique bending pattern seen in striated muscle. Thick filament contains myosin. Each myosin molecule has two heads attached to a single tail. The myosin heads bind ATP and actin and are involved in cross-bridge formation. Actin is the main component of the thin filament. Actin has a myosin binding site. When the muscle is at rest, the myosin binding sites are covered by tropomyosin so that actin and myosin cannot interact. In addition to actin, thin filament also contains several regulatory proteins, including tropomyosin and troponin. As I just said, tropomyosin covers the myosin binding sites at rest. If contraction is to occur, tropomyosin must be moved out of the way so that actin and myosin can meet. Troponin is the other major component in the thin filament. It's a globular 3-polypeptide protein. Troponin I, TNI, is an inhibitory subunit that binds to actin. Troponin T binds to tropomyosin and helps to position it on actin. The third one, troponin C, binds calcium ions and permits the interaction of actin and myosin. Both troponin and tropomyosin help control the myosin-actin interactions involved in contraction. Skeletal muscle fibers contain two sets of intracellular tubules that help regulate muscle contraction. Transverse tubules, T-tubules, are the sarcolemma of the muscle cell reaching deep into the cell interior. They are an extensive tubular network which is continuous with the extracellular space. T-tubules carry depolarization from the sarcolemma to the cell interior. The sarcoplasmic reticulum, SR, is an internal tubular structure. It stores calcium and releases it on demand when the muscle fiber is stimulated to contract. The SR regulates intracellular levels of the calcium ions. Okay, I have just introduced the complex microstructure of skeletal muscle. Then, before we talk about muscle contraction, I must give you a brief description of the neuromuscular junction, the site where the motor neuron terminates on the muscle. The axon terminal contains sacs of neurotransmitter acetylcholine, ACH. When the actin potential arrives, depolarization of the presynaptic terminal opens calcium channels. Calcium ions rush into the presynaptic terminal down its electrochemical gradient, inducing the release of ACH into the synaptic cleft. ACH then diffuses across the synaptic cleft and binds to its receptor on the sarcolemma. This produces a local change in the membrane potential called the end plate potential. We say the sarcolemma depolarizes. After completing its job, ACH is broken down in the synaptic cleft by acetylcholinesterase and diffuses away from the junction. Now comes to the mechanisms of muscle contraction: excitation, contraction, coupling. All parts of a muscle fiber I just mentioned and the electrical changes described in our previous lectures are involved in the contraction process. Binding of ACH to its receptor results in the polarization of the sarcolemma. The T-tubules then bring depolarization to the interior of the muscle cell. Depolarization stimulates the release of calcium ions from the sarcoplasmic reticulum. Calcium ions bind to the troponin-tropomyosin complex, which is moved away from the actin filaments. Myosin splits ATP to release its energy. Bridges on the myosin attach to the actin filaments and pull them towards the center of the sarcomere, thus making the sarcomere shorter. When all of the sarcomeres in the muscle fiber shorten, the entire muscle fiber contracts. The potassium ions leave the cell, restoring a positive charge outside and a negative charge inside, and the sarcolemma repolarizes. And then cholinesterase in the sarcolemma inactivates acetylcholine. Up to now, what I have described is a single muscle fiber contraction called a twitch in response to a single nerve impulse. It takes place in less than a second. No movement would occur if muscle fibers relax immediately after contracting. Normally, however, nerve impulses arrive in a continuous stream and produces a sustained contraction called a tetanus. During tetanus, muscle fibers remain contracted and are able to produce effective movements. In a muscle, such as the biceps brachial that flexes the forearm, an effective movement means that many thousands of muscle fibers are in tetanus, a sustained contraction. Subsequent nerve impulses would prolong contraction because more acetylcholine is released. When there are no further impulses, the muscle fiber will relax and return to its original length. So you see, even a very simple movement, such as blinking the eyes, involves a tremendously complex process. Many conditions may cause muscle contraction problems, such as low level of blood calcium, injured motor nerve, or stroke involving the frontal lobes of the cerebrum, etc. Myasthenia gravis is another example of a muscle contraction disorder. It is a neuromuscular disease characterized by fatigue and weakness of skeletal muscles. ACH receptors at neuromuscular junction are damaged by antibodies, resulting in difficulty muscle contractions. This is most likely to occur during repetitive activity, such as chewing or combing. Neo-stigmine can inhibit cholinesterase and decrease the degradation of ACH, thus enhancing and prolonging each end plate potential, reducing muscle weakness. This is the story with Ms. Lee. In summary, the action potential in skeletal muscle is carried to the cell interior by the T tubules. The polarization of T tubules releases calcium ions from the terminal cisternae of the nearby sarcoplasmic reticulum. Calcium ion binds to troponin C on the thin filament, causing a conformational change. Actin binds to myosin, cross-bridge cycling begins, producing contraction. Now we have completed physiology of the cells. The key concepts and principles include homeostasis, feedback regulation of body functions, the bioelectricity of body cells, mechanism of muscle contraction. From next week, we'll discuss the nervous system. See you next time. Thank you.